Hey, online, can you hear us?
Yes, we can.
Hello?
Yes.
Someone said yes. Okay. I should turn my volume on so I can hear what people are saying. And then
Well, I supposedly should hear it from the speakers, yeah? Because
Oh,
it's
yeah.
going to use that
okay, then I'll
speakers.
turn my volume off. So let me just see, because it has like the weird thing. So, oh, hey, that's good. I even get the presenter view. All right, that's great. That like never happens. All right. Great.
Nate in there.
Nate does not appear to be here yet. No.
You said.
Maybe I should share my, go ahead and share the thing.
Yeah,
Oh,
you should share
It says
your
that it's
screen.
disabled the screen share. Maybe I have to join with the other link.
I don't know what's you're doing. But
No, so it said it disabled the it said that screen sharing was disabled, so I think that I need to,
Hang on a second.
oh, for the Zoom, okay.
yeah,
It's not the sharing is disabled.
Oh, okay.
That's why he cannot.
Do you have just have to make them the code?
Yeah.
Okay.
okay, so you can just do that from there.
Okay, so you should.
Okay.
Share that screen.
So to here, and then do that. And then,
All right.
looks good.
Okay.
Hopefully people online see the, the presenter, or not the presenter view.
Yeah, I suppose they do. Yeah. Okay. I can check it.
Oh, yes.
Yeah, so what do you see? Do you-
Oh no, they see that.
They see the present, okay. So let's see, how do we fix this?
Swap display? No, but you can't do that.
Okay, let's,
Hey, hi. Good.
all right. Stop share.
Responsibilities.
Okay. So,
I quit last year.
there's something about the settings, right? Where it's like,
Yeah, no, it's, I mean, just like any kind of.
there's the display, something.
Oh, you should have display.
Yeah.
I'm like,
Display.
so are we.
Yeah, yeah.
If we have the same issue, I don't know what to tell
Okay,
you.
my settings are just.
It's just like bullying about it.
Settings. What? Please?
Sorry. No, it's related more when I,
So it's bad to watch.
yeah.
You know?
Maybe when you share screen, you can share the second screen. Is that possible?
Yeah, I can try that. Let me see. Okay, that's a very good idea. So let me do this. And then
Yeah.
let me go here. And let me share. Yeah, so share screen, share desktop too. Now,
Now, good.
okay. Like I
Such
said,
a fucking-
brilliant.
But just so you know, the security guard is really hassling the CMU students. It wasn't letting them in without an escort.
Okay,
I vouched for them, but if
Okay.
anyone's showing up now, like, no,
okay, do you want me to go back
Hmm.
upstairs? Wait,
that might be a good
Yeah.
idea.
okay,
Kelly.
sure.
Glad we can be a test.
Yeah, right.
Experience for
Right.
getting everyone into the building.
Going. Good. Yeah, so you got one more week? Are you?
Yeah.
Okay. Well, I'm still, I feel very sad that you don't have any time to go on your well-earned holiday.
Oh, it's fine. I'll hopefully there'll be more time in the future.
Yeah. Yeah. It's so hard to take time. Yeah, I also have never taken any time. I think I took 12 hours off between my PhD and postdoc, which was,
Yeah.
I went to see a movie, which was.
Yeah.
very nice.
Yeah, I mean, you gotta do what you gotta do.
Yeah.
We had our first vacation in like a very long time. Like vacation first in like 10 years. When we were moving.
Yeah.
So
That was a good one, right? You went to
yeah,
Hawaii?
exactly.
Yeah,
Just every time you change jobs, well, maybe sometimes
yeah,
it's a good time to take a vacation.
yeah. Just tell them that you're working remotely.
Right, yeah,
And yeah, just do it
You know?
from.
yeah. I can take a picture of the back of my room and
Yeah.
put it as the background.
Yeah.
That's awesome. You're the
You have
only
the
one.
beach behind you,
Yeah.
like, oh no, this is just
Yeah.
this is just one of those,
Yeah, I have
special backgrounds.
Yeah.
Oh, I have one of those like sound generators that sounds like the ocean.
Yeah.
It's not really.
Yeah, but that appears to be a real Mai Tai that you're doing.
Yeah.
Oh, wow, yeah.
It's just coffee.
You gotta relax.
Yeah. Cool, it's so fun. I think this is like the first live seminar I've been to. I've been to a real one.
We had a couple in the spring.
I guess yours, the job
Yeah.
in the last week.
Yeah, great. Yeah, I wasn't sure if I should do this on the, people are gonna come first. I'm gonna try.
Yeah.
See how it goes.
Oh, no,
People can always join by Zoom.
we're trying to bully all the external ones to,
It's nice, right, when you have a chance to, it's easier to like focus on.
Yeah.
That's good.
yeah, I'm so excited. It's
Getting back to,
yeah,
some things maybe are nice to have online, other stuff is nice when you can do it in person.
yeah. I suppose. I guess am I the master of ceremonies? I guess
I guess so.
Yeah. Okay, cool.
You can turn and when you start. Whenever it feels right
And
for
this
you.
mic is
This mic is
on,
live?
I think, let me see. Are we? Yes, I'm not muted, so and people can hear.
Okay, good.
That's a good question. To go to the. Where's the Zoom? I guess. Like it's. I can't see actually how many people are there. I think I would have to stop sharing.
Hi, John. All right,
Do you
ready?
want to step
Yeah.
over here? Yeah.
Yeah. All right. So if everybody's ready, let's get started. As I mentioned last week, the first couple of seminars this year we're devoting to our newly arrived Pitt faculty because we had such a wonderful recruiting year last year. And today it is my distinct pleasure to introduce John Barton. So John, John is off camera for those of you who are remote. So John did his PhD in physics at Rutgers University, where he studied with Joel Leibowitz. And after that, he moved to Boston to do a postdoc at MIT with Arup Chakraborty. And there he studied how HIV escapes CD8 T cells. After finishing his postdoc, he moved to UC Riverside to start his own group, where he was until where he started in 2018 and was until very, very recently, where we were very lucky to recruit him away to join us in last year's search. Pretty broadly speaking, his lab focuses on the coevolution of pathogens in the immune system, using temporal genomic data to try to build models of how they evolve over time, with a specific focus on HIV and SARS-CoV-2. And I think, thinking back to his job talk, it really was, I think, struck all of us right away how, exceptional he is in his clarity of thought and his ability to communicate really complex things. And so I think we're in for a real treat today with the seminar. So welcome, John.
Yeah. Thank you very much, Nate, for a really kind introduction. I'm very happy to be here in Pittsburgh and excited to share some of our recent work with you. So if you happen to see this, the talk when I came earlier in the year, a lot of these things are going to be familiar, but hopefully I'm also going to have a little bit of new things as well for people who might have seen that. So yeah, thank you very much. We'll go ahead and get started. So as Nate was mentioning, my group is really focused on understanding how pathogens evolve and how they interact with the immune system. So as you all know, viruses are able to mutate and replicate very, very rapidly, much faster than humans do, for example. And so this means that populations of viruses can accumulate lots and lots of genetic change in a short amount of time. And this is just one of a number of different reasons why viruses can be such an incredible threat to public health. And I'm going to start by giving you kind of a concrete example of this. I imagine probably many people in the audience, when I was growing up, I got the flu. And so as my body was resolving the infection, there were T cells and antibodies that were generated that were specific to the virus that I was infected by. And those are going to sort of lie ready and waiting if I get exposed to the same virus again to either cut short infection or even to prevent it entirely. But unfortunately, I didn't get flu just the one time. I got it multiple times over multiple years. And one of the reasons for that is that the virus that I was likely exposed to was not necessarily the same every year. And so here you can see like a quantitative example of that, which I'm drawing from some work from Trevor Bedford and collaborators from a couple of years ago. So this is looking at the evolution of flu viruses over the course of the last couple of decades, and in particular looking at this antigenic drift, so this change in viruses over time that changes how antibodies are able to respond to them. So on this vertical axis over here, you have this antigenic dimension, which basically tells you how similar these different viruses are from the perspective of the immune system. So if I have viruses that are very close together in that dimension, that means that they can be neutralized by the same set of antibodies. But as they get further and further apart along this dimension, it becomes harder and harder for antibodies that recognize one virus to recognize another one. And you can see this kind of consistent progression over time of even these different sort of subtypes of flu and how they're changing antigenically in terms of how they're able to be recognized by the immune system. So even if you develop good immune responses against one of these viruses, a few years later, you might be susceptible again. So this is one of the phenomena that we see, antigenic drift for flu. But of course, there are many, many other examples of this. In the last few years, we've seen different SARS-CoV-2 variants that have arisen, driven these new waves of infection. And later on today, too, I'll tell you about how HIV evolves even within individual infected people to escape from the immune system. So the goal of my lab is to really understand these processes in a quantitative way. And in particular, two of the main things that we're focusing on is trying to identify what are the mutations that are really critical for viral adaptation, which are the ones that really appear to be driving adaptation of the virus, and also developing quantitative models so we can try to predict dynamics of viral evolution in the future. And our hope is by doing this, we're first of all going to better understand the biology of these viruses, but then also be able to design better treatments or vaccines to fight against them. So that's kind of the broad goal that we're looking at. And in particular today, I want to tell you more about this sort of first point. So how can we use data to try to identify what are mutations that really appear to be driving virus evolution? And in particular, we're looking at this data of evolutionary histories. So we're interested in how changes at the genetic level will help a virus to replicate better or transmit better from person to person. And in general, that's something I'm going to refer to as fitness. So if a virus has high fitness, that's a virus that's able to replicate very well and transmit very well from one person to another. So that's what we're going to try to study. So just to give you a kind of a brief outline for today, first what I want to give you is a little overview of the methods that we've been developing to try to infer these fitness effects of different mutations from looking at the evolutionary history of a virus. And then I'm going to tell you about two different applications. One is to study how HIV evolves within individuals to escape from the immune system. And another example is looking at a different type of evolution, not within an individual, but across different individuals, seeing how SARS-CoV-2 has evolved over the last few years to become more and more transmissible. So that's sort of our outline for today. So first of all, the main thing I'm really focusing on here is understanding how how we can infer these fitness effects of different mutations for the virus from an evolutionary history. So what do I mean by that specifically? Let me start with a kind of a really simple example. So let's imagine that I'm looking at a population of viruses, and I have two different types of viruses. One is the wild type virus in gray, and then let's imagine that I have another virus, a mutant virus in red over here. And let's say that the wild type virus is susceptible to neutralization by antibodies, but the mutant virus is not. So if I was able to measure this or sample this population of viruses over time, one of the things I might see is that even if the mutants start out at really low frequency, because the wild type viruses are continually being neutralized by the antibodies, over time, I'm going to see the frequency of these mutants increase until at some later time point, maybe my population is totally dominated by mutants. And so I could look at this kind of data and I could see, well, this is a mutation that clearly appears to be beneficial, right? It helps the virus to replicate in this particular environment where there are antibodies that are eliminating the other types of viruses. And so I could even go further and try to analyze this and see exactly how fast does it increase in frequency to quantify this. So if I look at this kind of example, I might think that this is really easy, right? I can just look at the data, I see which ones are going to go up and which ones maybe go down, and that's going to allow me to determine what are the effects of these different mutations. However, unfortunately, when we look at real data is much more complicated than that, right? So I can't just look at this data and read off what the effects of the different mutations are. I really need some kind of method, some kind of organized way to try to infer these from the data that we have. So this is a particular example, a real example, looking at the frequency of different variants, different mutations within an individual who's infected by HIV over the course of several years. So I don't have just a single mutant to look at. I have thousands. In fact, about 6,500 over here. And to make things more complicated, these mutations are not, alone on their own background. So for example, if I have, a mutation at one site, I might see other mutations at different sites within the genetic sequence. And they could even be correlated with each other. So for example, if I have a mutation at this first site over here, G, I might be more likely to see another mutation at site 3, the C mutation here. So this correlation between different mutations is something that generally is referred to as like genetic linkage or linkage disequilibrium. So this makes things really complicated because now if I look at this, it's really hard to infer what the effect of an individual mutation is, because let's say I see something start at low frequencies and go to high frequencies. I don't know necessarily if that's because that individual mutation is really beneficial for the virus, or maybe it just happened to occur on a background of a bunch of other mutations that were really beneficial. And so it's just sort of hitchhiking to high frequencies. So we really need some kind of model to try to sort these effects out to figure out what the effects of individual mutations might be. So these models exist. You have standard population genetics models you can write down to do this. But the problem is that they tend to be very complicated. So past approaches that people have taken to this problem have often involved either just ignoring linkage entirely, so looking at each individual trajectory and trying to infer something from that alone and completely forgetting about the rest of the context, which obviously can be a problem, or things that are very computationally intensive that even if you have a few tens of mutations, it starts to become difficult to actually apply. So we need something that's more efficient than that. So that's basically what our group has worked on for some time. And our approach to that was, rather than trying to really innovate on the computational side, what we did is we started from these standard models of population genetics, and we used some ideas and some tools from physics to make sort of simplified models that turned out to be a little bit easier to actually fit to data. So we can actually write down, for example, what are the fitness effects of different mutations that are most likely to produce some particular set of data that we observe. And yes?
When you say mutations, by mutations, or are you breaking out like two mutations, like correlated?
Yeah, so I'm thinking about the effects of point mutations individually. In principle, you could think about combining things together, whether, usually we're working at like the nucleotide level, but you could also work at the level of amino acids or something like that. But typically, I'm just going to assume that, each individual, different nucleotide at different points in the genetic sequence can have a different effect on fitness. And I'm going to assume that those are just going to add together. So that's sort of the basic assumption. It's a really simple model, but it allows us to work with it in a simple way. Yes.
So did I understand correctly? So you're assuming that the effect of a single mutation eventually once it's translated, they sum up in terms of how it can escape from the antibody.
Yeah, so let me, and I realize I should repeat the question for the people who are online. So the question is, we're assuming that the effects of individual mutations, those can sum up to tell us, for example, how you get escape from antibodies or some other thing that's important biologically for the virus. Yes, so that's our assumption for now. In principle, you can have all kinds of other complicated things that and maybe I can comment a little bit on that way towards the end. But yes, it's a very important question. Yeah, so this is our goal. So I want to tell you just a tiny bit about this, and then I'm going to show you some examples and simulations and move on to look at real data. Okay, so here's sort of the basic idea. So we start with one of these standard model from population genetics known as the Wright-Fisher model. It tells us about evolving populations of individuals as they undergo mutation, recombination, and also selection. And let's see, I'm going to check. I don't know if I can do this. Okay, great. Just checking the chat really quickly. Chat was saying people appreciate having things repeated. Great, I will continue to do that. It's looking at how populations evolve subject to mutation, recombination, and selection over time in discrete generations. So these dynamics at the level of genotypes, so full genetic sequences, can be quite complicated, but I can also project those dynamics down on the frequencies of individual variants or mutations. So here you can think about these as like different nucleotides at different sites. So for the ease of presentation here, I'm just going to assume that at each site we either have a wild type nucleotide or a mutant, and I'm not going to distinguish between different nucleotides, but you could easily expand this to consider either different nucleotides or different amino acids at your genetic sequence. So these dynamics are complicated, but you can project it down to a dynamics on these mutant frequencies. And this follows what's called a Fokker-Planck equation. You don't have to know what that is. I'm going to tell you basically what this means. And it has two different terms. So one term is called the drift term. And so this tells us about the expected change in these mutant frequencies over time. And so, if I can, there we go. So the drift term looks like this. I'm going to explain what these different parameters are. So first, there's a selection coefficient for each site. So that tells us what the fitness effect is of a mutation at site I. If that's positive, that means the mutation is beneficial. If it's negative, that means the mutation is deleterious and harms replication. So that's the first variable that you see here. I also have a mutant frequency. So this is how many individuals in my population at a particular time have mutation at site I. And what also appears here is a paramutant frequency. So how many individuals in the population have mutation at both sites I and J at the same time. So this might look a little bit complicated, but really the interpretation of these different terms is pretty intuitive. So the first term is a term that's just telling us about natural selection. So this says, for example, that if I have a mutation at site I that's beneficial, it's going to tend to increase in frequency over time. But how much it increases in frequency depends a little bit on what the frequency is right now. So for example, if I have no mutants in the population, they can't increase in frequency. They need to be generated through mutations first. And if all of my population is composed of mutants, so the X is equal to 1, I also can't increase in frequency anymore, right? So this is just telling us about natural selection. The next term is a little bit more complicated. This is related to the linkage that I was talking about earlier. We might call this term linked selection. So I think it's easiest to explain what this term does by giving you a little example here. So let's imagine I have mutations at two sites, at sites I and J in some genetic sequence. And let's imagine that mutation at site I is completely neutral. It has no effect whatsoever on fitness. But mutation at site J is beneficial. So if mutations at sites I and J tend to appear together on the same genetic sequence more often than you would expect by chance. So in other words, if that term in the parentheses there is positive, then I'm still going to expect the number of individuals that have mutations at site I to increase in frequency over time, even though that mutation does absolutely nothing, just because it happens to be associated with another mutation that's beneficial. So the fate of a particular mutation depends not only on its individual effect, but also on the rest of the genetic background where it appears. So that's what that term is telling us about. How do we expect things to change over time? And then the next term is a what's called a diffusion term. So this characterizes just fluctuations in these frequencies due to the randomness of replication alone. And so for people who have a background in population genetics or evolutionary biology, this is actually exactly the same thing, this covariance matrix of mutant frequencies. This is the linkage disequilibrium matrix, D. It's just scaled. Yes.
Understand this, because that's gonna make a huge difference for me and you.
Sure.
When we say XJ and
Yeah.
XIJ, so when we have XJ, does it mean actually under this frequency, some part is XIJ? Does it make sense what I'm asking? So I'll be like, when we say XJ, we are just looking at all, anytime we see a J there, and all the others could be other mutations.
Yeah, so the question is basically about the definition of the XIJ and the XJ. So to clarify, let's say we're looking at an individual XI or an XJ. Basically, I just take my population, I take all the genetic sequences I have for each individual. I count how often you have a mutation at a particular site. That's your XI. So it doesn't matter what other mutations it has. That's just averaging over everything. And then the XIJ, now I look at all the individuals in the population, and I only count them if they have a mutation at both I and J.
So XIJ is then a subpopulation of XJ.
Yeah, exactly. So XIJ is going to be, yeah, strictly less than XI or XJ. Yes. Oh, let's see, you mean down here in the main selection part. So this is, let's see. So the question is whether there should be a square.
Their frequencies not count, right?
Yes, these are frequencies. That's right. That's right. So these are sort of similar in terms of the, if you think about the units, these are similar because the XIJ is also a frequency as well. And so this correlation is going to range from, well, the range is going to be a little bit restricted compared to the other ones, but the range, the XIJ could also go, let's say, from 0 to 1. And so that whole correlation term could be negative and could be positive. Yeah, but can't go all the way to -1.
XI times XJ would be that happening random. Yeah.
Exactly. So that's why, yeah. So this is basically looking at how tightly correlated mutations at site I and J are. So the comment from the audience was that if it was totally random, if there's no association between mutation at site I and site J, then the probability of getting them both together would just be the product of those individual probabilities, or in other words, XI times XJ. So this is telling you how correlated those are. Yeah. Okay. So those are the terms that we have here. And then we also have, for this term that's telling us about the randomness of fluctuations due to stochasticity of replication, I also divide by the population size, the total number of individuals that I have. If I have a big population, my fluctuations are small. If I have a small population, my fluctuations are big, a lot of randomness. Okay, so my math is almost done. I promise. So basically, what we can do then is we can take this equation, this Fokker-Planck equation, and we can transform it using some ideas from statistical physics to what's called a path integral. So this is really just a fancy word for telling us about the probability of an entire evolutionary history. So I can write down what's the likelihood of generating some particular evolutionary history as a function of the selection coefficients. So in other words, the fitness effects of mutations, that's what I want to find. And without telling you the details, once we've made these kind of simplifying assumptions, I can actually find out analytically, I can write down an expression for the selection coefficients that are most likely to have generated a particular piece, a particular set of data. So I just want to mention here that we are not the first ones to have used this kind of path integral approach to study models population genetics. I first learned about this from Ville Mustonen and Michael Lassig's work back in 2010. But as far as I know, we were the first ones really to apply this specifically to inference and trying to understand what these parameters might be like from data. So I'm going to spare you the details there. And instead, I'm just going to give you an example and simulation to show that you can really do this, and this approach works pretty well. So here I'm going to start by giving you an example of a simulation. So we simulated the Wright-Fisher process with a population where I have some mutations that are beneficial, some are neutral, some are deleterious. And this you can think of as like a very simple example of the kind of complicated dynamics that we observe in real populations. So when you look at this, we might tend to think that, okay, the mutations that end up at high frequencies, those are ones that are probably more beneficial. The ones that end up at low frequencies, probably more deleterious. The neutral ones, it's a little bit harder to say, right? But I would say it's not obvious to just look at this and pick out which ones are beneficial, deleterious, and neutral. But we can do that with the model that we have. So here, for example, we can infer what's the selection coefficient for these beneficial mutations. So we can identify those from this kind of data. And this, again, might not be too surprising. So these beneficial ones tend to either stay at high frequencies for a long time, or they start at low frequencies and go to high frequencies. So, fairly makes sense here. We can also identify which are the mutations that are deleterious. And again, this is kind of reasonable because they tend to either go from high frequencies to low frequencies or stay at low frequencies for a long time. But because we look at how things are correlated together, we can also perhaps surprisingly infer what are the mutations that are really neutral, that don't have any effect on fitness at all. And this is maybe a little bit surprising because if you look at those trajectories, some of those trajectories for these individual mutant frequencies look very much like the ones that were beneficial or deleterious before. And the reason that we're able to sort those out is because we look at how things are correlated with each other. And we can explain, for example, that the reason that one of these trajectories goes to high frequencies is because it happens to appear on a beneficial background. That's a better explanation of the data that we observe rather than that mutation individually being strongly selected. So we get all of this from these estimates that we can write down analytically. So let me just check here really quickly. Question in, okay, that's a direct question. So I'll save that one for later. This is just to show you that this method at least works in principle. And so, we can demonstrate this on simulations, but how can we use this to learn about the evolution of real viral populations? So let me go then to, tell you about a couple of these examples. So one of the first examples that we studied was looking at how HIV evolves, within an individual to escape from the immune system. So I'm going to start by just telling you a little bit about how HIV interacts with the immune system to kind of give you an idea of what we might be looking for. So when cells become infected with HIV, as for other viruses, the genetic material of the virus is released and leads to the production of new virions that can go to infect more cells. And then through these sort of normal processes of the cell, some of these viral proteins will get broken up into short peptides. They're about 10 amino acids long. They're presented on the surface of the infected cell, together also with self peptides. And these can be recognized by T cells. These short pieces of viral material, they're called epitopes. So if the T cell has a specific T cell receptor that binds to the epitope, can detect that this is foreign material, and trigger an immune response that kills the infected cell. And it's known that this is one of the main ways by which HIV infection is controlled in individuals. So T cells play a major role here. But of course, that's not all. In the adaptive immune system, we also have B cells that produce antibodies. These antibodies can bind to and neutralize virus that's either free-floating or that's bound to infected cells. So we have these different arms of the immune system that are both working together to control infection. But we want to understand really what is the relative pressure of these different arms of the immune system and how is it different in the way that it applies to the virus. And in particular here, one of the issues that we have, and one of the reasons why HIV is not able to be controlled by the immune system in almost all cases, is that if the virus develops mutations within these epitopes, so either within T cell epitopes or within parts of the virus that are bound to by antibodies, that can then prevent the immune system, either the T cell receptors or antibodies, from binding to the mutated form of the virus, allowing it to escape from immune control. And this really happens within individual patients. So we want to understand, what are really the forces that are driving this. So to do this, we looked at a population or a data set of 13 different individuals who were followed from the time point of initial infection up to several years after. And these were individuals who were not on antiretroviral treatment during that time. So we really see sort of the natural interplay between the immune system of these individuals and the virus. And so one of the first things that we observed when we looked at this data is something that's well known in the HIV area, which is that pressure from these T cells that I was talking about before is really a major driver of the evolution of the virus. So if you look across the HIV genome in these individuals, roughly 2% of all possible mutations that the virus could make are in regions that are targeted by T cells. So they lie within T cell epitopes. But if you look at mutations that are most strongly selected, so in the top couple of percent across these patients, about 45% of them actually lie within these T cell epitopes. So it's more than 20-fold enhancement compared to what you would expect by chance. And it turns out that taking this correlation or this linkage into account is really important to reveal this. So if you don't, you only find about half as many of these strongly beneficial mutations as you would otherwise. And so, why is that? The reason for that is that when the virus generates these escape mutations to escape from the immune system, it often doesn't just generate one mutation, it generates multiple ones, and those can compete for dominance in the viral population. So here's a really simple example looking at one epitope in one of these patients. And on top, I have the frequency of these different escape mutations that the virus generates within the epitope. And then horizontally is just the time in days. So this is a very simple example, but you can see competition between these variants that affect their dynamics. So if we use our approach to try to estimate what's the effects of these different mutations, we would infer all of these to be beneficial. They aid the virus, they help its replication by allowing it to escape from the immune system. But if we were to ignore this linkage, this covariance between different mutations, we actually wouldn't infer the same picture. So because we ignore how they're competing with each other, the selection that we estimate is a lot smaller than we would otherwise, and we only infer positive selection. We only infer the mutations are beneficial if we see them at some finite frequency at the final time point. All these mutations that appear transiently and then go away are not inferred to be beneficial at all. And that doesn't really seem to make sense in the context of immune escape here. So this is an example we see for just one, a single epitope. But one of the things that's nice about this approach is that it scales really well to larger amounts of data. We can consider many mutations at once. And in particular, in this set of data, we have sequences from half genomes of HIV. So I can expand this, for example, to look at the sort of landscape of selection that we infer within a particular individual. So this is the same individual as the epitope that I was showing you before, across half of the HIV genome. So that's arranged in a circle here, starting from Paul and going all the way around to the 3' LTR. So this is the later end of the sequence. And you can see these dots that are around these circles are the effects of individual mutations that we infer. Ones that are in red are beneficial mutations. They help the virus. Ones that are in blue are mutations we infer to be deleterious. And the bigger those dots are, the stronger those effects are. So this is the picture that we see. One of the things you also observe is I have these like boxes, right, that are around this circle. And so those represent CD8 T cell epitopes that we have from experimental data. We know that those are T cell epitopes that are targeted by the immune system. So you can see here just really obviously, if I look where the big red dots are, they lie within the T cell epitopes. So there's clearly a lot of pressure on the virus to escape from these T cells. And one of the things I think is, yes.
So these epitopes, of course, are also mutating and they mutate in and out, or probably out.
Yes.
Right.
Right.
So at some point, So how do you deal with the? I mean, is that timeline also in the
Yeah.
model?
Yeah, so this is something we're looking at right now. And I have plots, but I don't have them on this thing. So one of the interesting things is that we do expect that really there should be some effect of, that these pressures are not really constant in time. Here we've assumed that they are, but really they're not. Because, for example, one thing that happens, that doesn't happen very often, but happens sometimes, is that you have a T cell response that's generated very early in infection, and then the virus quickly makes escape mutations that basically allow it to, no longer be targeted by those T cells.
Right.
And then if you look a little bit further on, now the T cells have no targets. They never see their epitope anymore.
Right.
So all the T cells that responded to that epitope go away.
Yeah.
And then the virus, sometimes the mutations that it makes to escape from T cells might be deleterious in another context. So those mutations can revert, actually, can go away in time. So one of the things we're looking at, and it turns out that we can actually do, is we can actually estimate selection pressures over time. And so, for example, when we have these kind of cases that are pretty rare, but do appear to, do actually happen in this data, where you have these reversions, you can really see how the selection pressure, rises early on and then falls late as the pressure to escape from the immune system goes away. So this is something that's still very much in progress, but here we've assumed that everything is static, and I think is in most cases is not like a crazy assumption, but yes, we want to get to that point where we can really look at things over time.
Right. So overall, these are not accumulated mutations in this region. So it's just the individual mutations, in different individuals.
So the question is, are these like accumulated mutations? And this is actually just one patient.
Yeah.
So this is a single individual, and not all of those mutations that you see are actually going to fix or going to, let's say, remain polymorph, but a good fraction of them actually will. So it will accumulate quite a fair number of mutations over the course of, a year to several years of infection. Yeah, let's see, other questions? Oh,
Just to
yeah.
make sure I am following along. So you need time course data.
Yes,
And
that's
then
right.
what you do is you fit the dynamics equations where you define the drift and
The fluctuation term, right. Mhm.
and from that, built into that is this S. That,
Yeah,
yeah, okay.
exactly. Yeah, so
So I
just,
understood it. Okay.
yes.
I didn't ask a question.
No, yeah, so just clarifying, basically one, for the people on Zoom, the idea here is we have this dynamical data. We're looking at how we have really in data measurements of how the viral population over time, how the viral population is changing over time. And we're trying to infer these model parameters that best explain, based on this model I showed before, that best explain the observed evolution of the population. So that's what I'm showing here. So yes.
Sorry, I think a couple more clarifications.
Yeah, of course.
So then I think in the previous slide you talked about, like if you ignore the covariance, so there you're talking about if you ignore the SIJ, like co-selection term, or you're talking about if you ignore the fact that in the population, multiple variants are arising at the same time, they have to compete against one another.
Yeah.
That it was the latter that you were referring to.
Yeah, that's right. So in the previous, so the question is, what essentially is the linkage term that we could be ignoring that we were, that I was mentioning before. And it's exactly this kind of correlation term. So what happens, I'll just show you really quickly. I'll go back and then go forward. So here, these mutations that you see on top in the epitope, those arise mostly on completely distinct backgrounds. So you don't see different escape mutations arising in the same virus. There are multiple subpopulations of viruses that have different escape mutations, and those all compete with each other. So, if I put that in terms of this sort of like XIJ term, all those XIJs are 0. But then the product of Xi and Xj is not 0. So I have this additional term here. It's basically saying that because there's competition, I don't actually see frequencies increase as much as I would expect them to, because now, for example, this top mutation that you see here has to compete with a bunch of other beneficial mutants. And so that's how we get these kind of adjusted estimates of the effects of different mutations.
And there's one more clarification. I'm curious about.
Yeah.
Do you get measures of epistasis out of it? It's like if the SIJ is much larger than the SI over SJ, is that like the signature you would?
Yeah, so the question is, can we get estimates of epistasis out of this? And we're very much thinking about this. So it turns out to be a little bit more complicated because now we have to look at kind of higher order correlations, but we can do that. And one of my postdocs actually here in the audience is working on exactly this question. So we're trying to extend to these things and also figuring out, how you can estimate this without having to have like really, really huge amounts of data that have lots and lots of correlations. But yeah, in principle, we can look for exactly those sorts of signatures. Yeah. Okay. So let me continue on here for the moment. So, I just want to mention here, one of the interesting things that we see also is we see interference, not just between escape mutants within a single epitope, but also even in between different epitopes. You can see escape mutants for different epitopes that actually compete with each other. So this is another thing that sort of really emphasizes this strength of selection for escape and how these different escape mutants are competing in a viral population. So one of the things that we observe here is you see readily this selection for escape from T cells, but for antibodies, it's not really quite as obvious. And so if we wanted to look for that, what we should do is we should look for mutations in the surface protein, the envelope protein, which I've highlighted here in yellow, and that would be where mutations to escape from antibodies would occur. And so we do see some mutations in the envelope that are beneficial, but they don't appear to be quite as strong as the ones that we see within T cell epitopes. But this is a little bit difficult to say because here we don't know exactly what parts of the envelope protein are being targeted by antibodies in this particular person. So to try to look into that in a little bit more detail, what we did is we actually studied an individual who develops what are called broadly neutralizing antibodies. These are antibodies that are able to neutralize many, many different strains of HIV, unlike typical antibodies that are produced by individuals infected by HIV, who are mostly specific for the virus that they're infected with. So these are really of a lot of interest to people in the HIV vaccine area because those are the kind of antibodies that you want to make to have an effective vaccine that's going to protect against many, many different strains of HIV. So we looked at a particular individual, CAP256, and this individual develops a family of broadly neutralizing antibodies called VRC26. And when we look at the picture of selection in this individual, we see something that looks like this. And here is the VRC26 epitope, so the part of the envelope protein that's bound to by the antibody in this particular person. And so one of the things you see here immediately is that it looks a lot more complicated than the case I showed you before. Part of this is that there's a lot longer data, really detailed data here to infer more subtle effects. But then also this individual ends up having what's called superinfection. So they're actually infected by multiple different strains of HIV at different times that recombine with each other very extensively. So this creates really, really complicated dynamics in terms of the evolution of the virus within this individual. So it becomes really critical to correct or to account for this linkage because if you ignore that, actually the mutations you would infer to be the most beneficial are actually even synonymous. So they don't even affect protein coding. So if you don't look at how things are correlated with each other, you really have trouble identifying any kind of signal here. So I want to turn back to this VRC26 epitope. So that's where I should see mutations that could potentially help me to escape from neutralization by the antibodies. And so if we zoom into that and look at things in a little bit more detail, here you can see the effects of different mutations within that epitope. And I want to focus on a couple that we know are associated with resistance to that lineage of antibodies. One is right here in codon 165. So this appears to be deleterious, but this is actually selection against a mutation that makes the virus more sensitive to VRC26. So this is one mutation that increases or selection against the mutation that makes it more sensitive. So this is selection for resistance. I also have a bunch of mutations over here in codon 169, where multiple different mutations are associated with resistance to VRC26 lineage antibodies. But these are really the ones that are sort of known to affect resistance, and the total strength is not really that high. So the fitness advantage of those mutants is estimated to be like around 1% or so. There is a mutation that we see that appears to be pretty strongly selected, that one over here in codon 162, but this is actually an interesting story. So this is a mutation that's selected very, very early on in the evolution of the virus within this individual. And this actually changes the pattern of glycosylation of the envelope protein that makes it more sensitive to VRC26 lineage antibodies. So this rises before those antibodies ever actually appear. So one of the things that we really have observed in this particular example, and also when we look across patients, is that the signature for escape from antibody responses appears to be a lot more subtle, a lot sort of softer than the signal that we see for T-cell escape. And this is something that we're still trying to look into in more detail now to really understand this process of not just evolution of the virus, but really co-evolution between the virus and antibodies, especially in individuals who make broadly neutralizing antibodies, to really understand that process better and perhaps help to connect to efforts to develop vaccines to elicit these broadly neutralizing antibodies. Okay. So I told you now about how we can look at the viruses evolving within individuals in the, yes.
So, John, if it is a broadly neutralizing antibody, right, you can go and check what region of the envelope that is, and can you just immediately tell that the reason why it's a broadly neutralizing antibody is because it's disrupting a major aspect of the infection, say the interaction with the cell.
Yeah. So wait.
Can you tell that as well? It seems to me that this analysis should be looked in the context of the fact that this is not an arbitrary site, that it's something that must be functionally.
Yeah. Yeah, right. So the question is, let's say let's say you have an antibody. Can you sort of look at where it binds? Which finding that in the first place can be a little bit difficult because you don't know necessarily, because these epitopes are sort of conformational, so it can be a little bit hard to determine. But say you determine where exactly this is binding, can you tell just from that information, for example, whether it's going to be a broadly neutralizing antibody or not? So this, I wish I had more information. I'm sure there are people who are more on the antibody side who could tell you more. I think one of the things that's sort of interesting, I think, about this is that you do tend to see, so the antibodies that are broadly neutralizing, as you would expect, they tend to bind to parts of the envelope that are like more conserved and more functionally important. They bind to the CD4 binding site, they bind to conserved glycans, things like that. But that's not sort of always the case. You also have CD4 binding site antibodies that are not broad. So just based on the region where they're binding, you can't necessarily tell if they're going to be broad or not. So I think that's a very interesting question. I think an unresolved question of, can you tell, for example, how broad or how potent a particular antibody is going to be, for example, from structure or analysis of the binding site alone. But I think, yeah, we would love to and are trying to get into analyzing these things together. So looking at how the antibodies are really the evolution of antibodies that are targeting a particular area, and then also the evolution of the virus. So that's something that we haven't gotten into yet, but we really want to, and want to understand this in a lot more detail. Okay, so I want to shift now to something that's a little bit different. And rather than looking at evolution within a particular individual, I want to think a little bit about evolution of a virus actually across different individuals. And this is something that I mentioned like in my example early on about evolution of flu, but of course this also has happened for SARS-CoV-2 in recent years. So how can we understand this and how can we describe this process? So in principle, we really need a different model to address this. We can't look at evolution of a population, let's say within a person, to describe this. So instead, we need a model of epidemiological dynamics of virus transmission from one person to another. So we started to build such a model, and basically we used a branching process model where we consider the dynamics of fractions of individuals who are infected by different variants of a virus. assuming that they can also have mutations. Those mutations affect how the virus is transmitted. And in this, we also account for super spreading of the virus. This is something that we know happens in SARS-CoV-2. And the idea of this is that basically, if I have a, let's say a more typical infection process, then the distribution of new infections that's generated by one individual who's infected by a virus might be something sort of well-controlled, like a Poisson distribution. So if I infect, let's say on average, one more person, and I have this kind of standard distribution, sometimes I infect one, sometimes two, sometimes none, right? I get something that's kind of tight and well-controlled. With super spreading, even if the average number of new infections is the same, I tend to get more shifted distributions where I have a lot more zeros and then a lot more big, infection events. So maybe sometimes I infect nobody, sometimes I infect nobody, and sometimes I infect 24 people. So we're also accounting for this kind of thing in our dynamics. So I'm going to skip all of the details and just tell you that somewhat surprisingly, and we were certainly surprised to see this, if you make kind of similar assumptions in terms of, assuming these transmission effects of individual mutations are not too big, assuming they're additive, you actually get dynamics that looks very much like the Wright-Fisher model, except these drift and diffusion terms I told you about earlier are rescaled. And they're rescaled, for example, according to how much super spreading there is. So if there's a lot of super spreading, the diffusion term, for example, becomes very large. So the stochasticity is really big just due to the super spreading. So what this means in particular is that I can solve for the transmission advantage of different mutations the same as I did before, just with this extra rescaling, the same as we did for evolution within an individual. And so we did that. We analyzed data from GISAID, which is an enormous repository of data originally for viruses like flu, but now, of course, for SARS-CoV-2. When I talked about this last time, we were using data from August of last year. Now we're using it through June of 2022. So we have 5.6 million sequences across 126 different regions. So those could be like states or small countries, things like that. So this is 4 times more data than what we had just earlier this year. And we used that to try to estimate the effects of different mutations on SARS-CoV-2 transmission. And the story that we found now after Omicron, so our previous data was pre-Omicron, looks very similar, but with the addition of some very impactful Omicron mutations. So these are the effects of SARS-CoV-2 mutations on transmission that we infer across the genome, again, arranged in a circle with the meaning of these things, the same as what I was showing you before for HIV. And so one of the things that, of course, you see when you look at this is that this huge cluster of mutations that are beneficial for viral transmission in the spike protein, and especially in the S1 subunit of spike. And if I look at the most impactful mutations, the ones that we would infer to affect transmission the most, these include things like the Delta 142 mutation, so that deletes a particular residue that's in the N-terminal domain. And that's an Omicron mutation shown to affect escape from antibodies. But then I also have a lot of things that are not in principle related to immune evasion. For example, these mutations in the receptor binding domain that are changing how the spike protein binds to the ACE2 receptor, how it fuses with the membrane, and then also these mutations in this furin cleavage site, which affects how the spike protein is cleaved into these 2 functional units. So we see this really readily in the data. But I think one thing that's interesting. So of course it's nice to be able to highlight these mutations for which there's a lot of experimental support. One of the things we also observe is there are clusters of beneficial mutations in other regions outside the spike protein that appear to be really impactful. So those include, for example, deletions in sp6 and some mutations in the nucleocapsid protein. So I think one of the things that's potentially interesting, contribution of this work is we can kind of highlight these mutations that might not be in the most obvious areas, but ones that might be really important for the biology of the virus that would be good things to study experimentally to understand why they might appear to have these effects on transmission. So in addition to looking at these individual effects We can also add up the effects together to estimate the total change in transmission for different versions, different variants of the virus. And so this is the picture that I showed last time, for people who were here back in February. And now with the addition of a bunch of new data, the picture actually doesn't change very much, which is very comforting to me as someone who does inference, that if you add a bunch of data and things change wildly, it gets a little bit scary. But here it looks very, very similar. The only change is alpha kind of slides down just a little bit, delta slides up a little bit more, which makes sense because our previous data was from sort of the middle of the delta wave. So we see here, if we look at our sort of previous generation of variants, delta was really very dominant. But alpha and some others were not too far behind. Delta we would estimate to be roughly twice as transmissible as ancestral SARS-CoV-2. But unfortunately, Omicron and sort of related subvariants have really changed the game. So if we look at these more emerging variants here, we see that Omicron itself looked to be about 3 times as transmissible as ancestral SARS-CoV-2. And now if you look at things that now have become very dominant, even since June, something like BA5 is almost 4 times as far as we would estimate. So you have these really enormous effects.
That's the booster, right?
Yeah, this is in principle. So get your booster. This is a very good thing. Always good to mention, and get the flu vaccine at the same time. So unfortunately we see these really big effects. So just in the last couple of minutes, I'm going to tell you about something really briefly, which is one thing we might be really concerned about specifically in the case of SARS-CoV-2, but perhaps also applications to flu. We want to know, what are the strains of this virus that are going to be really dominant in the future. So we wanted to be able to identify really early on if you have sets of mutations that appear to make the virus a lot more transmissible, whether that's due to immune escape or other factors. And so can we do this? So to do that, what we did is we looked at we estimated the selection, so the change in transmission for all sets of linked groups of mutations. So for example, like the alpha mutations, they all come together. We would consider that to be like one linked group. So we looked at this, first of all, globally, so in all the data that we have, and then in every individual region at each intermediate time up until this cutoff over here. And I want to tell you sort of why are we doing this? So we're doing this because we know that, if I look, for example, at the frequency of different variants, let's say in Pittsburgh, there are going to be some fluctuations up and down with finite sampling, right? I don't want to look at, some little jump in frequency and then suddenly say, oh no, there's something, crazy new that has come out. So we want to be able to identify things that could potentially be dangerous, could potentially contribute a lot to changes in transmission without overreacting to noise. And so we did this up until this cutoff time of February 2021, so like roughly halfway until the data is out, so that we really have, we're using one part of our data to sort of train a model and another part to predict and test. So what we found is that if I look at linked groups of mutations that are estimated to change transmission by 10% or less in global data, they're never inferred to have selection coefficients higher than 13% in any region at any time. So if I see any group of mutations that has a coefficient that's stronger than that in aggregate, that means that I can say with really high confidence that those are going to increase transmission by, let's say, at least 10%. And that's something you might say is sort of a rough threshold for something being concerning. Though, of course, one can always argue about where exactly to draw any kind of continuous line, right? But roughly, we're going to use that for our threshold. So if we do that and we apply the...
What do you define this coefficient? 10%, because of course, you just need a little advantage for that, to differentiate this eventually in time. Right?
Yeah.
That time dependent.
Oh, sure. Yeah. So the question is,
What does it mean, like 10%? Like after a month of the right.
So that means sort of like, so the question is, what does 10% really mean? So this is we should think about this not as like a frequency sort of thing, but this is in terms of how many the spread of infection of different variants. So if something has like selection coefficient of 10% relative to something else. So it's, its fitness is like 10% higher. What we would say is like the R value, the expected number of new infections is also going to be 10% higher. So that's what we mean. So it's like a 10% advantage per like infection cycle. Yeah. So that's our rough kind of choice for the threshold. And if we apply this automatically to data, so not trying to sort out, not trying to tell manually what is alpha, what is delta, but just looking for things that are correlated and seem to be strongly selected, we overwhelmingly identify well-known variants of concern. So alpha, delta, Omicron, different Omicron subvariants. That's something like 98% of the things that we detect. And the other couple of percent are also arguably ones that increase transmission. So it looks like you're overwhelmingly able to really reliably identify things that people know or have identified as variants of concern through other means. And importantly, we can find those really early on. So I just want to show a couple of examples. One, for example, is the rise of alpha in London. So, it was originally seen not exactly in this area, but this is one area for which there's data where look. And back in 2020, we would infer alpha to be significantly beneficial, the set of alpha mutations, by November 12th. So even before it was designated as a variant of interest or concern. So you already see strong selection, even when the frequency of that variance is quite low, around the 1 to 2% range.
During your training?
So this would be, yes, so this would be during the training period. Let's go to things that would not be in that period. Delta in Great Britain, for example, so this is now after that in 2021, we would be able to identify as, really strongly beneficial, having a coefficient of at least 10% by April 15th. And then Omicron, in principle, using data even back to 24th of October from South Africa. Of course, one of the issues that you run into here, though, from a practical perspective is it can always be difficult to identify these things in real time because sometimes it takes a long time for things to actually get uploaded to databases and all of that stuff. So the reporting delays are obviously going to slow that down. But I think the main point to be made here is that in principle, if you have that information there, even when things are at very, very low frequencies in the population, you can already identify that these might be concerning. So that I think is potentially valuable. So, okay, we're out of time, for sure out of time. So I'm going to go to my summary over here and just tell you briefly, so what we did is we developed this fast, even analytical method that we can use to study both in within and between host evolution. We saw these different contrasting patterns of immune-driven selection for escape from T cells versus antibodies in HIV evolution. And when we looked at SARS-CoV-2, we saw these clusters of beneficial mutations in different regions. We can also add together to estimate transmission benefit for whole variants. And it appears to be really sensitive enough to give us this kind of early detection or early warning about more transmissible variants, which I think potentially could be something that's valuable. So I want to thank my collaborators here, including students, Brian and Liz, who did a lot of work on the SARS-CoV-2 thing in particular. A bunch of other people who are involved in extending this in various ways, looking at selection, or sorry, looking at epistasis, looking at how selection might vary in time. I want to thank also my collaborators, Matt McKay and his group, who are now at the Doherty Institute in Melbourne, and also funders. And we've had some questions so far, but I'd be happy to take any additional questions that you have. Yeah. Right.
Someone answered the question. First of all, this was an amazing presentation, and it actually answered a lot of things that I always have, myself asking about. So, actually, I have a question, and it's gonna sound like it's, but it's not, because what you did here is really amazing regarding SARS-CoV-2. So, what I've seen in the literature and also my own studies is that, and thanks God it's actually not happening, is that the mutations on the RBD domain are not additive. If it were, we were in a very bad situation right now in terms of the COVID pandemic. But it seems like
What do you
some,
mean additive for what? Additive.
like, for example, when you look at the antibody binding site. Okay, so one mutation, for example, decreases the antibody binding or increases the ACE2 binding affinity. And you know that another one is also increasing the ACE2 binding affinity. But if these two are combined, actually they have a lower, it can be that they have a lower ACE2 binding affinity. It's proven actually in the literature, experimental implementation.
Yeah,
So just to-
that happens for any in general.
Yes, but here, but we also wonder that some mutations are combining in SARS-CoV-2 so that we have a higher infectivity. And I was always wondering that, and now there's a model. I mean, you are doing that, and it's like, what's the take-home message here? Like when you look, you have all this data. I don't have all this data. Like mutations in different proteins are combining, or in the same protein from different domains. Like you have all this data.
Yeah.
What is the message here?
Yeah, so the question to repeat for people on Zoom is basically, there's you observe that there is, let's say you look at mutations in the receptor binding domain, you look at experiments, those don't necessarily add together completely additively, where if I have, one mutation that increases binding, another mutation that increases binding, if you put them together, the change in binding is not necessarily exactly the same as if you would just sum those individual changes. So sort of, how do you interpret that? If I'm, saying this correctly, and then also how do you interpret, for example, combination of different mutations within the receptor binding domain, but maybe in other places or in other proteins. So I think, this is an interesting question, and I think certainly, so there are a couple of things. First, the assumption of additivity is of course an assumption, and might not be true. I think one thing that would be really interesting to look at is to look at and we're trying to get to the point where we can do this, look at models of epistasis and see, for example, if you can see evidence for collections of mutations that have effects that are not purely additive. So that I think is something we want to try to interrogate in the data and see if we can find evidence for that. It can be, one part I think is tricky though, because even, we're ultimately looking at changes in transmission, which might be different from really molecular level changes, like changes in binding and things like that. So in some sense, even though, I think we can certainly validate by, we hope to find, and we do find mutations that we infer to be really important have effects that have been known in measured experiments. But how those add in terms of transmission, I think is even yet another question. And I think that's one that you can sort of never answer ethically, right? You can't do transmission studies of SARS-CoV-2 to try to get that effect. So I think that's very difficult to isolate, but it's interesting. These things might not be exactly the same. But I think also certainly, if you just look at the evolution of the variants and forget all about the models over here, you do see mutations arising in many different regions, not just in the receptor binding domain, but also in this N-terminal domain, in regions that are, in these other proteins like these NSP6 deletions. So I think that to some extent, all of these things are adding together to increase transmissibility by kind of tweaking different specific things. Better receptor binding, more efficient cleavage of the spike protein, better evasion of past antibodies. All of those things I think are adding together to lead to greater transmission. So
there was nothing that like standed out, because I'm actually
yeah. So
I'm really curious. I mean, this is the first time I've seen such a study, and I'm very excited about what you did. So I mean, like RBD and like NBD, like do they like combine, for example, those mutations that
So the question here also is, okay, what about, can you kind of break this down more into categories, and how do these things come together quantitatively? And that's an interesting question. We have looked at this sort of individually. So we kind of looked at our top mutations and try to understand what's the evidence for those, where are they, all of those things, but we haven't done a comprehensive study of, like, for example, X% are in the RBD or, something like that. So that's a very interesting suggestion too, would be interesting to look at. Let me go to one question here in the chat, because I don't want to ignore the people on Zoom as well, if that's okay. So there's a question from Rahil Sethi for looking at SARS-CoV-2. Is the data collected globally? So we're looking at, for example, at Delta. But in other places, we saw in April 15th, we would identify it in Great Britain. But in other places like in Southeast Asia, it was already, really established, which is, indeed very true. Wouldn't it make sense to look at data globally for dominant variant prediction to control infection? So that's true. So we do look at data globally, but we definitely, we sort of divide this up into multiple regions, and we assume that the regions are sort of quasi-independent, but that selection is sort of common between them. And we use all the individual regions to estimate some kind of global coefficient. So the reason that we break things up into regions in the first place is that sampling is uneven between different areas. So of course, for SARS-CoV-2, as people who know a lot of this might know, for example, the UK has done just an absolutely tremendous amount of sequencing. I think more than half of the sequences collected globally are from the UK. That doesn't mean that more than half of SARS-CoV-2 sequences, SARS-CoV-2 infections globally have happened in the UK, right? So we want to use information from different regions without letting the regions that are really well sampled just overwhelm our results. Then also there's some kind of isolation between different regions. Certainly, there are questions about how well people can mix even within a country, but the infections are going to be much more, let's say, self-contained within the UK than between, if I look at like Europe as a whole or something like that. So this is another reason to break things up into regions. For analyzing new variants, we're looking at each region individually. For that particular question about emergence of Delta, I wish that we had great data, but the data that we have, for example, from India has tended to, and in some other nations, sometimes it comes in big spikes, so we don't really have good time resolution. So basically, we don't really feel confident that we can apply our approach if the data, if the sampling is too sparse. So even if you have a lot of sequences, you do need to have good time resolution. And so to the extent that we have that, we can look at other regions. But in this particular example for emergence of Delta, UK, or in this case, actually Great Britain, so not including Northern Ireland, that's one where we had ample data for the emergence of that particular variant, and we could actually, we could apply our approach there. So that's why we use this sort of as an example. Yeah, Nate.
So I really like this idea of the sort of like the microscopic model and accounting for like bursting dynamics. The evolution of the virus. And I was sort of curious if, once you break that down, can you find evidence of viruses that are exploiting sort of different reproductive strategies? Like you can just imagine one that has like a comparatively low, selection coefficient, but has a very large burst size. So it's sort of like infected frequently, but when it does, it builds jackpots. Like then break down different categories. Yeah, so the question is about, looking at this kind of microscopic models of you have the super spreading dynamics and things like that, and potentially, the comment from Nate is that we can imagine viruses could exploit this. In addition to changing, let's say, the average number of new infections, you could also change, through certain, let's say, changes in the virus, you could affect your propensity for super spreading or something like that. And of course, this is also like a complicated interplay between really virus biology and like human behavior and things like that as well, right? But I think potentially you could have things like that. And one of the first things actually that we want to look into that we haven't done yet is there's been some estimated changes in the amount of time that it takes for an individual who's infected with one variant of the virus to actually pass it on. So what you would call like the serial interval, how much time between, so if I get infected and I pass it to someone else, that takes on average X number of days. And the idea is that this has actually maybe changed a little bit over the course of the pandemic, that for ancestral SARS-CoV-2, maybe this is like 5 days, 6 days, something like that.
but for something like Omicron, maybe it's like 4 days. So in this case, actually, even if the average number of infections is, let's say, not quite as high or not as much as you might expect, just because the generations are happening faster, you might see those dominant, those viruses become dominant. Or you could imagine in different circumstances, maybe a slower cycle could be dominant. So I think there's a whole kind of ecology there to think about. But the first step, I think, for us would be looking at, let's say, like the generation time, trying to understand how that would be a feature. But potentially this could, expand to a lot of different things. Yeah, it's really interesting kind of dynamics to consider. So, is there? Oh, okay. Wait, what? Can you?
Someone raise their hand.
Oh, raised hands. Okay, I can't see raised hands. So if you have a question, please just speak. And I hope we'll hear you.
John, I have a question.
Yes, I can hear you. Great.
That was great. I really enjoyed it very much. So I have a general question. In the method that you developed, the Fokker-Planck method, as far as I understand, you predict the probability or the frequency of occurrence of a mutation at a given site or correlated mutation, but you don't know what is the type of mutation. Is that correct?
So that's sort of what I showed here for the simple, just for the purpose of illustration. But in principle, so in the actual model that we use, we're really tracking specific nucleotides, or in principle, we could also break this down into at the amino acid. So we do have that information, but here just for then you get more indices and it becomes a little bit more complicated. So just for the purposes of
I see.
presentation.
Also, otherwise, for example, you could do that for the spike, all possible amino acid substitutions.
That's right.
Great.
Yeah, and at the moment, we've actually worked at the nucleotide level specifically to understand, for example, there could be even mutations that are synonymous mutations that might have an effect. Also for validation, because if we were to say find that a bunch of synonymous mutations were inferred to be really beneficial, we would be in trouble. So thankfully we don't infer that. But we actually, for example, we have 1 or 2 examples that have some experimental validation where you might see a synonymous mutation that has some kind of effect on transmission. So, but we could also, yes, we could certainly look at this at like amino acid level.
Yeah, that's great. Thank you so much. I enjoyed it a lot. I thought this talk was here. I didn't realize that, but I enjoyed it very much.
Thank you. Okay, yes, so we'll take a,
That was actually, I had 2 questions, I'm so sorry.
Yeah.
So when you showed the HIV results, you showed that all the mutations were like happening at the T cell epitope, right? So I always wondered myself, and I came actually to a point to ask this question after your presentation. I wasn't able to formulate it.
Yeah.
So the reason HIV or some viruses are worse than others, is it the reason that they have a higher probability to mutate at this location?
Oh, so the question here is, we see escape mutations within T cell epitopes for HIV. Is this one of the things, is this really like a reason why, for example, HIV is so difficult to deal with compared to other viruses? And I think the answer is that there's certainly that contributes, but also is not necessarily the whole, answer. So let's say we look at HIV. HIV also infects immune cells. So just the process of infection degrades the immune response. This is a big deal. HIV can also can be latent, so it can sort of stay inactive in the cells that it infects and then reactivate later. So this is also the reason why, for example, drug treatment is able to suppress infection but not cure it. So it certainly is. It is. I think one of the main reasons why immune control is very difficult in HIV, but is not necessarily the only reason behind. There are many other features also that contribute to various, the sort of pathogenicity of different viruses. Yeah, so I think maybe if anyone has other questions, please feel free to contact me. Happy to chat at any time, especially here being fresh to Pittsburgh and really excited to talk with people. Ask me about the technical part. I actually cannot shut up about it. So I will, be very happy to tell you anything you'd like to know. And thank you everyone for coming. I was, very much enjoyed it. All right.